Hello and welcome to this next module in the MIDD training series, where we're focusing on model-informed decision-making in drug development. My name is Mark Gasronguay. I am the founder and CEO at Metrum Research Group, and this particular module will focus on the simple question of why use models to inform decisions. Right up front, I want to acknowledge several individuals and organizations that are responsible for the content in this presentation. They include the scientists at Metrum Research Group, the MIDD Training Steering Committee, and all other collaborators across industry, government, and academia that have influenced my thinking and are responsible for some of the examples that we'll present today. A little bit about me. I'm trained in pharmacy with a professional degree, PhD in pharmacology, and a postdoctoral fellowship in pharmacometrics at the FDA. I've been working in this field for several years and dedicated almost the last 3 decades to the growth of the science of pharmacometrics, working in industry, academia, contract services, and nonprofit sectors. I'm passionate about applying quantitative modeling and simulation, in particular to those medical problems where there are unmet needs. And I'm also a strong proponent of open science. Our organization shares a lot in this way through open source tools, open model, and open courseware. Probably most importantly, I'm grateful at Metrum Research Group to work with a fantastic team here and all of our collaborators. I've also been privileged to have mentored and learned from more than 25 graduate students and fellows in my role as graduate and adjunct faculty. I'll save the rest of the details for later. These slides are included, and you can dig into these recognitions and publications at your leisure. We'll pick up on Emerson's point of view here as we deliver the content in this part of the workshop. Really, what I'd like to focus on are the principles that underlie model-informed drug development. There are many methods, lots of tools, algorithms, and software potentially that could be applied to this set of principles we call model-informed drug development. For our training today, let's focus on those principles. So in this module, we will run through several topics, starting with a brief overview of model-informed drug development. The key focus today will be on this question of why use models when we may have observed data. And it really comes down to this concept of theory versus empiricism. We'll talk about how models can be used to derive insights around unstudied conditions. We'll then transition to the application of model-informed drug development in a decision-making role, reviewing some opportunities in drug development, acknowledging that many drug development decisions are made based on estimation of model parameters. But what we're talking about here is something that goes broader than that uses model development and applied simulation to explore a variety of decision paths. And then we'll wrap up with a review of some benefits and challenges. So what does model-informed drug development mean to you? This is a term that's been used quite frequently in many venues, and it's actually evolved over the years from model-based drug development to model-informed drug development, to model-informed drug discovery and development, and many variations on that theme. You might have some preconceived notion as to what that means. When we speak about MIDD in this course, we'll talk about it as a way to consider the use of models to support drug development and regulatory decision-making, and also to develop a quantitative theoretical understanding of disease and therapeutics. By models, we mean mathematical models. They can be simple descriptive models, They can be statistical models that describe a variety of distributions and hierarchies in that sampling distribution. The models can attempt to be semi-mechanistic, describing physiology and pharmacology, or they may be more physiologically based, truer to anatomical and physiologic processes, or any combination of the above. MIDD actually has a long history, maybe not in that term, but modeling and simulation has been applied in drug development for quite some time. More recently, you'll see this particular timeline is relevant. Peck and others described the use of model-based methods for therapeutic drug monitoring, population pharmacokinetics and pharmacodynamics, and the potential to apply that sort of understanding to drug development questions. Lou Scheiner and others promoted the idea of this learn, confirm paradigm where drug development or clinical pharmacology is a process of experimentation represented by mathematical model-based theories and then further confirmation with experimental data. In the 2000s, we've seen rapid growth of these methods, including things like exposure response, disease progression models, clinical trial simulations, and model-based meta-analyses as almost a routine exercise in drug development. Rick Lalonde and others described the value and application of these approaches in a review article in 2007. More recently, we see the mainstream application of mechanistic models, physiologically based pharmacokinetics, quantitative systems pharmacology models to this entire suite of tools, methods, and principles. One thing that I do acknowledge is that our discipline does use a lot of jargon, and I'm trying to be careful here to define terms the first time we introduce them, but you'll often see the acronyms used subsequently. Hopefully, that's not too difficult to follow. In a 2015 paper in CPT, Pharmacometrics and Systems Pharmacology, the elements of model-informed drug development, or MID3, drug discovery and development, were outlined. And they include the kinds of things that we would typically aim to learn about in drug development. First of all, a mechanistic understanding of the drug action and also the drug disposition. Human pharmacokinetics, absorption, distribution, metabolism, and excretion, the so-called ADME properties. Use of models to compare candidates, whether internal lead versus backup, or maybe even comparing to external standard of care. Models to evaluate study designs, both preclinically and in clinical trials. Models can be used to help facilitate the risk-benefit characterization or to make early projections of that risk-benefit ratio at early development time periods. The selection of dose and dosing interval, dosing schedule is something where model-based approaches is typically used, as is the application of these methods and principles to select individualized or specialized dosing for patient populations that may be unique. Bridging across populations, whether that's populations of different patient types or studies in healthy volunteers to studies in patients, can also be accomplished using model-informed drug development principles. Well, all of those learnings could potentially be observed by conducting experiments, clinical trials, and observing the resulting data. So why build models when we can simply observe experimental data? I'll address that question by reviewing a seminal paper in the literature. This is a key publication by Lou Scheiner, a commentary on the clinical pharmacology and the choice between theory and empiricism. This was published in December of 1989 in Clinical Pharmacology and Therapeutics, just happened to be my first year in a PhD program in pharmacology. Really, when I read this paper, it opened my eyes to a new way to think about science, at least new for me. The key here is an understanding of the role that theory plays in the design and collection of data and subsequent improvement of that theory. So I'll take you through the paper and a key example, and hopefully that will help to illustrate why those of us who are proponents of model-informed drug development believe that modeling does add value beyond what can be observed in empirical data. The particular case in question had to do with nicotine pharmacokinetics and pharmacodynamics. It was known at the time, and this is some key work by Porcher and others in 1987 and subsequent years, that nicotine exhibits some sort of tolerance. And in fact, if you were to look at the plot of the pharmacodynamics, in this case here viewed as the change in heart rate from baseline versus blood nicotine concentration, the typical time course of this plot illustrates a clockwise loop, sometimes known as a clockwise hysteresis loop, or maybe a proteresis loop. This relationship is indicative of a non-constant concentration response relationship. And in fact, if you look at it carefully, you see that initially concentrations rise on the x-axis and the response rises. That makes sense. But then over time, even as concentrations increase, the response is muted and diminished relative to the response seen at very similar concentrations earlier on in the infusion. There were multiple potential explanations for this, but two that were proposed were, one, this is due to distributional disequilibrium, or in other words, the fact that the sampling site for nicotine concentrations in the blood is not kinetically in sync with the site of action. That blood nicotine does not represent the concentration of nicotine at the site of action, and that they're temporarily disconnected. Another potential explanation is that there is some sort of pharmacodynamic acute tolerance. In other words, the system becomes desensitized over time in some way. That's another potential explanation for why the response at later time points at similar concentrations is lower than the response at the same concentrations earlier in the infusion. The series of experiments and theory in the form of models that were applied to this question provide us a nice illustration of why model-informed drug development is so useful. Let's start with the theory. So the initial explanation was a potential distributional disequilibrium. On the left-hand side here, you see a graphic of a physiologically based pharmacokinetic model. You've seen this before in the training modules. I'm using it here not because it's a mathematical model, but it allows us to conceptually think about the infusion of drug, the observation of concentrations and response in physiologic context. Our drug here is nicotine, and it's being infused intravenously. Well, that infusion site is actually not in the central pool of venous blood. It's usually in some other venous access point that rapidly flows to the lung and distribution through the heart and arterial blood. So if we imagine an infusion of nicotine here, intravenous infusion, passing through the heart and lung circulation, reaching arterial blood, and now perfusing all of the tissues in the body. You see the nicotine then travels rapidly to sites of action. In this case, we consider the brain the site of action for nicotine's effect on heart rate. This is a centrally mediated effect, at least in part. And so that's why we've identified PD here as a pharmacodynamic endpoint with the brain compartment. It's not until after nicotine has reached the site of action that it appears in the pooled venous blood compartment where PK sampling is taken. So you can see that kinetically, even though this may happen over the course of just a few minutes, the distribution of nicotine is distinct across these anatomic compartments. That understanding of a potential distributional disequilibrium between the sampling site, venous blood, and the site of action, the brain, was used to construct an experiment. Here you see on the right-hand side a pharmacokinetic study conducted in rabbits in which drug was infused intravenously, nicotine in this case, and was sampled in a variety of locations. First, the arterial blood was sampled in the femoral artery. That's indicated by the upside down red triangle, and also denoted here in the graph. There was also a sampling site in the internal jugular vein, represented by the blue triangle here. That's the blood that's immediately effluent from the brain. And then there was a femoral vein sampling site for the pooled venous blood nicotine concentration, indicated by the yellow square. Now you see that over time, all of these compartments tend to track together with respect to the concentration time course. But early on in those periods of the first few minutes post-infusion, there are some dramatic differences in the concentration of nicotine at these different sampling sites. The arterial concentration is the highest, as you might expect. The jugular vein, which is the effluent blood from the brain, has a secondary concentration here, which is lower than the arterial concentration because of tissue extraction of drug across the brain, but still much greater than the venous pool that we see in the yellow square in the femoral vein. Well, this is an example of how a theoretical conceptual understanding can lead to an experimental design, which then further elucidates potential mechanisms describing the initial phenomenon of that clockwise hysteresis loop between heart rate and blood concentration of nicotine. Now, a potential explanation for that relationship is proposed in terms of a mathematical model. On the top right, you see that mathematical model here. It's a simple one, not as complex as the physiologic structure I just showed you. We have infusion into some arterial concentration, and that has some sort of distribution to a venous sampling site. But similarly, there's also some distribution to a site of action or concentration at the effect site, CE, which we are postulating is the brain for nicotine's effects on heart rate. It's that concentration that drives effect. Whereas in the prior plot, it was the concentration in venous blood that was used to predict the response. Now we can use this theoretical understanding to simulate or to estimate what the brain concentration might be. And when you plot that relationship, heart rate versus the simulated brain concentration, the characteristic clockwise hysteresis loop is gone. And you have this collapsed relationship that looks more like a typical concentration response relationship. One conclusion might be that, well, that's because the site of action is now driving a response, and it's a concentration at the site of action that results in some expected concentration response relationship. But that was just one of our potential explanations. Remember, we talked about this phenomenon of distributional disequilibrium, the effect site equilibrating more rapidly than the sampling site. But we also postulated another potential explanation, And that was the development of acute tolerance or desensitization of the system. So the authors considered that second potential explanation and asked themselves, how could we develop an experiment that would help us to distinguish between the two phenomena? Using this model, they derived an experiment which allowed for the separation of the distributional effects from potential acute pharmacodynamic tolerance by adding another infusion to the experiment. So in the top right, you'll see an example here where you see blood nicotine concentration in 2 infusions rising and falling in the second infusion, starting here at about 120 minutes. These are 30-minute infusions, and allowing that concentration to fall off. The interesting thing about this design, the really clever part, was that it was postulated that if you were able to space these infusions over time, there could be some insight gained about anything that would be due to an acute tolerance in the response. So by doing so, they proposed an experimental design that still maintained 2 infusions, but now spaced them at different intervals over time. You'll see here increasing all the way out here to several hours. These experiments are done in healthy volunteers, and at the same time that the concentration time profile is being sampled in plasma, sampled from a venous access point, heart rate is being measured. The interesting thing here is that the heart rate response is not predictable across each of the different experimental designs. In fact, if you contrast the short interval between the 2 doses, you see that the first dose rises to a peak heart rate that is greater than the peak at the second dose, whereas the concentration profile shows a maximum concentration achieved at the second dose that's higher than the first infusion. However, as that repeat dose interval is spaced out, that particular effect is diminished, and you see that by the time the doses are spaced even further, the second infusion results in a heart rate response that is equal to or greater than the first dose pharmacodynamic response. Shown on the bottom left here are the clockwise hysteresis loops for the 2 infusions under each of those designs, with panel A reflecting the short interval, panel B the intermediate interval, and panel C the more distantly spaced time interval. One thing that you'll notice is that the hysteresis loops here, the clockwise hysteresis loops, look very different across these experimental designs, where clearly the 2 doses in experimental design A are separated in the rising and falling of concentrations over time. By the time you get to design C, you almost see a superimposable loop. Now, this might intuitively lead you to conclude that there's more than just a distributional disequilibrium going on here. It could be that experimental design C reflects the distributional disconnect, but the earlier experiments, or the experiments with shorter intervals between doses, exhibit something more. There's more than just distributional disequilibrium going on here. And so that particular study design, which was informed by some theoretical understanding, leads to a new theoretical understanding. And in fact, if we apply the initial model here, which is the one that describes the distributional disequilibrium principle, to one of these repeat dose data sets, you'll see this model is really unable to capture the pharmacodynamic response. Heart rate is indicated by the open circles here, whereas concentration in venous blood is the dashed line. That's the model predicted. And the model predicted pharmacodynamics, the model predicted heart rate, is the solid line. Well, with this distributional disequilibrium model, you see that clearly, although the second dose pharmacodynamics are rather well described by the model, the first dose with the peak heart rate effect is not. This model does not allow the accommodation of the additional potential pharmacodynamic tolerance that's going on. All this allows for is the distributional disconnect. So what do we do when the model doesn't match up with the data? We consider the theory and revise it accordingly. And that's what was done here. And the same model structure with an addition now, where some tolerance driver was added to the model here. Here it's viewed as a first order compartment being driven by the concentration at the effect site. But whatever the true mechanism for tolerance, it's something that is related to the response at the site of action and actually diminishes the response over time. With that sort of theory applied as a model, the top right panel here indicates a much better description of the heart rate time relationship. So here we see that theory is improved by experimental data, resulting in a revised theory. Now, to take that further, we can consider applying the model to new questions. In the actual sequence of events here, additional experiments were conducted studying the pharmacokinetics of nicotine after nicotine gum, as well as the intravenous infusions. And that information was used to augment the pharmacokinetic model to describe the pharmacokinetics of nicotine after administration via gum versus intravenous infusion. Taken together then, a new question was asked. Why does nicotine gum not help very much with cigarette abstinence, particularly first thing in the morning? You'll note in the plot on the left that we have a time course here which indicates the heart rate versus time after a cigarette or an infusion versus the heart rate after ingestion of nicotine via gum. This is actually the application of the theory as a simulation. And you can really say that model-based simulations are applied theory. The question being addressed here is what happens in the morning after that abstinence from smoking overnight, and how does the nicotine gum compare to the cigarette? Heart rate response after smoking is much greater and rises much more rapidly than it does after chewing gum, which might indicate why the gum is not so effective if, for example, heart rate is reflective of those effects that manifest abstinence from nicotine. However, over time, you see that the gum does catch up a bit to the smoking of cigarettes and maintains more of a steady state concentration. This is a simple example, but it illustrates the opportunity now to take a theory that's been challenged and improved with data and now apply it as a mathematical model to conditions that were previously unstudied. That allows us to generate insights and drive decisions and hypotheses for future experiments. So the concepts here are best summarized by Shiner himself in the discussion section of the 1989 paper on theory versus empiricism. He says, thus by going beyond empiricism and stressing understanding, not data collection, we not only answer our first question, but we also gain far more. For clinical pharmacology, as for all other sciences, the goal is theory, not data. The pursuit of theory involves both induction, imagination, and deduction, with subsequent empiric verification. One is useless without the other. If theory is kept as the goal, it will not only be more aesthetically satisfying, but will inevitably lead to insights and technique applicable far beyond the original locus of study. In short, and in truth, there is nothing so practical as a good theory. So with that in mind, let's move forward now to consider how we use that theoretical understanding of the system to inform decisions that are made in drug development. Lalonde et al. Summed this up nicely in their 2007 article. We should view drug development as a model building exercise, during which we continuously update our knowledge about a new compound and use this knowledge to inform our decision-making and drug development strategy. This point of view is completely analogous with the theory and data iterations that Shiner described. Ideally, MIDD approaches would integrate across multiple levels. Imagine you're on a development team developing compound B1, which has multiple clinical trials ongoing or planned. Well, at the same time, there may be another compound, A1, with a different mechanism of action in development for the same indication, and maybe a backup with the same mechanism of action as B1, this one called B2, in development for multiple indications. You can see that in this sort of scenario, there's lots of reliable prior information and knowledge that can be applied to the competing or follow-on compounds. Information about the disease state, the patient population, key endpoints. But there's also information available about the probability of each of these programs leading to successful new therapeutics. And those sort of considerations can be also taken into account. With multiple compounds having intersecting mechanisms of actions and indications and other competing development programs, it can be beneficial to develop MIDD plans at multiple levels. Those levels could include the particular compound or individual development program, maybe all of those compounds with similar mechanisms, or compounds in a particular indication. Those might be vetted, though, across a broader therapeutic area versus other indications in that therapeutic area. And then similarly, different therapeutic areas might be balanced across the full R&D portfolio for a company. An understanding of the drug action and impact on disease is necessary for all of these types of decisions, but it goes further in that there will be questions about probability of success in translation from non-clinical to clinical studies, probability of achieving target expectations for efficacy and safety, probabilities about the availability of the patient population to generate an adequate sample size for a new clinical trial, and also the relative probabilities of success with respect to therapies across different therapeutic areas in the portfolio. There are many opportunities to apply MIDD principles in drug development. Here's a schematic of some of those activities and principles that you might apply in a single development program, starting at the preclinical research phase all the way through market access. For example, in the preclinical space, where maybe the goal is to identify the development candidate molecules, methods using quantitative systems pharmacology, physiologically based pharmacokinetics, and biomarker exposure response might be helpful in identifying which of those compounds are more likely to be successful development candidates. When that decision's been made and now the new therapeutic moves on to the translational development or preclinical development phase, where the understanding of exposure response with respect to biomarker, toxicity, pharmacokinetics, and mechanisms to translate this preclinical knowledge to establish a first-in-human dose are necessary. Model-based methods play an important role. Once successfully initiating a new IND, compounds moving into clinical development can also benefit from the prior modeling work conducted in preclinical research and translational development, but also now to consider the goals of the phase 1 clinical studies, where an understanding of human pharmacokinetics, tolerability, the relationship between exposure response for tolerability or maybe biomarkers, and early assessments of how this molecule might shape up versus potential competitors, looking at model-based meta-analyses from competitor published data, and other model-based assessments of the probability of success. Transitioning from phase 1 into phase 2 in clinical development, the goals shift from that understanding of human exposure and tolerability to really thinking about the proof of concept that this new therapeutic will provide efficacy and safety in the intended patient population. So questions about which doses to be studied in early phase 2 studies, how to design those trials to generate sufficient understanding and knowledge to warrant further investment in the clinical program. Methods like population PK/PD, estimation and simulation are relevant here. Exposure response to support dose selection, and trial simulation methods, clinical trial simulation methods that will take the phase 2 data and now establish the basis for the confirmatory trials in phase 3. In phase 3, these studies are conducted, and oftentimes data are collected with the specific purpose of applying model-based methods to inform things like support of efficacy and safety conclusions, dose selections, dose adjustments in special populations for regulatory filing purposes. On the regulatory filing, model-based methods are also used during the regulatory review cycle. Whether it's reviewing the submitted modeling and simulation content provided by sponsors, or maybe it's an attempt to gain additional insight by pooling data across internal submitted data and prior experience held within the regulatory agency. Model-based methods can be applied to guide decisions and support the regulatory review progress. And then when it comes to market access, post-approval, questions such as comparative effectiveness and some idea of the real-world efficacy given real-world data are important with negotiations with third-party payers and placement on formularies and state-sponsored medical systems. Those goals can be supported using some of the same models and data that were used during the clinical development program, but also borrowing from electronic medical records or metadata from competing therapies. Now that just outlines some of the activities that could be conducted with respect to a single development program. There are other activities that really are most well suited to an investment across a therapeutic area or a broader platform of compounds. Things like building disease progression models, models of quantitative systems pharmacology, competitor meta-analysis, and trial simulation tools for particular indications and patient populations. Those are places where model-based methods actively play a role. However, you may be familiar with related decision-making based on estimation of parameters that you could say come from a model. And I'm going to highlight some of those here and contrast that with a full MIDD type approach to decision-making. So many times, inferences and decisions in drug development are driven by estimated quantities, where a study is designed to focus on a particular question and generate some empirical data that could be used to answer that question. The data are summarized as a parameter and oftentimes a parameter and point estimate with confidence intervals, really describing that particular comparison of interest. A theoretical understanding of the system and how things work underneath the hood is not really the goal here. It's just primarily to make an assessment of a comparison of some sort. And although these are sometimes model-based or simple models, but the inferences are limited to the specific conditions studied. They're predefined and limited to that point, and really focused on the quantities estimated. So let's look at a few examples of that sort of inference or decision making based on model-based estimates of a particular parameter derived from a very specific study. One example of this parameter estimate derived decision making is bioequivalence. In bioequivalence, a specific study is designed to compare the pharmacokinetics of a test formulation versus the reference formulation. Summaries of the pharmacokinetic profile are calculated, such as the area under the curve or the maximum concentration, Cmax, and those are compared between the test and the reference formulations. The comparison is a ratio, and if there were no difference between the two, you would expect a ratio of 1. However, it's not sufficient to just simply have a point estimate that arrives at the null value or the point of no difference. We also consider the precision around that by representing a 90% confidence interval of the ratio of test to reference for the particular PK parameter. In the bottom right plot, you see that ratio illustrated for a couple of different scenarios. First, at the top is the brand name or reference situation. And then the next three are for different iterations of a test product. The first is a non-equivalent test product in that the point estimate is lower than the expected value of 1, but the true criterion here is that the lower bound of the confidence interval extends below the region of equivalence. And in this case, the region of equivalence is for the log transform ratios to be between 0.8 and 1.25 relative to the reference formulation. Again, you see a similar scenario with a non-equivalent formulation that's too high in the next line. And then the last example is one that is bioequivalent in that the interval for the relative ratio is contained within the region of bioequivalence. This is one of those cases where a model-derived parameter estimate, in this case it's a ratio of test to reference AUCs, is viewed in terms of a point estimate and confidence interval to drive a decision. Now that's a model-based guidance, but it doesn't necessarily go beyond that particular comparison. Similarly, you might consider those kinds of comparisons made with respect to comparing two treatments. Here's a meta-analysis of two different types of antiviral therapies. In red is the triple therapy, and in blue is a two-drug combination. Similar to bioequivalence, our null value or point of equivalence is a hazard ratio of 1. Any of the point estimates and intervals that fall to the right of that favor the triple therapy. Any of the intervals and point estimates that fall to the left of that favor the two-drug combination. Again, this is a simple model-based estimate, point estimate and confidence interval that can be used to guide decisions. It can be used not only to determine something that might be statistically significant, but also to have some idea of the magnitude of the improvement in a triple therapy, in this case, versus a two-drug combination. Still, that's the extent of the inferences drawn here. In population pharmacokinetics, we can also derive similar estimated quantities when exploring the impact of individual patient covariate effects on summary measures of exposure in this case. On the left-hand panel is an analysis that describes the impact of a variety of different cut points in individual specific covariates: weight, creatinine clearance, liver enzymes, bilirubin, and then the categorical of race and sex. That's viewed relative to a reference AUC for the typical individual. That's an AUC steady state here, denoted by the dashed line. And the gray region here reflects a similar concept to that bioequivalence, 0.8 to 1.25 region. The model is used to estimate the covariate effect magnitude and its 90% confidence interval. In this case, the model does allow us to do so at a variety of different cut points, even if those were not observed in the data. So there is an added benefit. But it's still largely an estimation-driven inference. On the right-hand side, the same sort of plot is illustrated for another summary metric here. In this case, it's Cmax at steady state. So those methods of parameter estimation-driven decisions or inferences are quite common in drug development. They can be model-based, but they are limited to the particular comparison that is defined. The more complete MIDD approaches actually build that theoretical understanding of the system via a mathematical model and then use that model as a dynamic simulation tool. It's theory in application to explore a variety of different scenarios and conditions. That's the kind of model-informed drug development that we're talking about in MIDD. It's interesting that that particular approach is also quite relevant in mitigating some of the challenges in decision-making that a development team might face. In fact, the literature of the psychology of decision-making, cognitive biases, and related complications is very rich. But there are a few features here that I'll highlight, which are well suited to being mitigated by using model-informed drug development principles. One of those is that in a group, when decisions are being made, imagine the group is a development team, the decision performance is usually not as good for the group as a whole as it would be for the most informed individual. Unfortunately, that's difficult to know who the most informed individual is when you have a team of experts working together. That's why transparency, sharing of knowledge, and understanding the system is key to making solid decisions and will improve group decisions. It's also been shown that organizations with formal decision processes and structures make better decisions. Formal processes are necessary when you're applying model-informed drug development principles to making decisions. Precision and consistency with the way that decision-making is key. It's been found that inconsistency in an organization and how they make decisions is potentially a bigger problem than some of the cognitive biases that are inherent in our intuitive type decision-making. Again, a planned model-informed drug development strategy applied across an organization brings that sort of consistency to the problem. We know that there are multiple sources of cognitive bias that impact our intuition-based expert decisions, and that's why expert decisions should be viewed versus more objective assessments of the problem by transparently laying out assumptions, prior knowledge, knowledge gaps. We'll talk about all of that in the following module. And then intuition is not the best way to make decisions, as you might expect. It's been shown that group decisions are more well informed by running scenarios of possible outcomes. Objective, data-driven exploration of scenarios has been shown to improve decision-making performance. And that's exactly the kind of thing that we do in a model-informed drug development strategy, exploring scenarios and possible decision paths through mathematical simulation. So you might think that MIDD is purely mathematical, but it does connect very relevantly with psychology, the psychology of decision making. What we want to get to, if you recall, is this application of theory to gain new insights. So although we have the mechanisms to gain insights or make decisions based on parameter point estimates with a specific study design, what we want to be able to do is take this theoretical understanding in the form of a mathematical model and apply it to new questions, just as we reviewed with respect to the comparison between nicotine, gum, and cigarette smoking and the impact on the pharmacodynamic response. In order to do this effectively for new therapeutics, the understanding of the dose-exposure response relationship is critical. I know you've heard a lot about that in the prior modules. I want to just emphasize that it is relevant here as we apply these models for making decisions. That relationship, or those relationships, apply to safety and efficacy, maybe across multiple endpoints. And they inform things like dose and regimen selection, patient population identification, and maybe dose adjustments necessary in special populations, the design of the clinical trial itself, and how that feeds back in your understanding of the dose-exposure response relationship, provides a controlled and rigorous link to real-world data, and maybe assists in making that sort of an extrapolation combined potentially with some real-world evidence from electronic medical records or other sources. And it also establishes a strong framework for the new therapeutic when comparing the projected efficacy, safety, or whatever response endpoint you're interested in to those of competitors when viewing them across a competitive landscape. So how do we apply that quantitative theoretical understanding to model-informed decision-making? There is a framework you could apply, an MIDD strategy that I'll outline here and that we'll see applied over and over again in the subsequent training module. First, before we're able to make any decisions, we have to understand the questions. And those questions should be specified in a way that anybody could understand, but also in a very specific quantitative manner so that the features of the endpoints or decision criteria are understood to a sufficient degree that a model-based approach could be directed towards that. Those questions could be questions about the probability of attaining the target product profile, the probability that a dose or treatment regimen will meet expectations, the probability that a trial design will result in a successful trial, questions about the populations and dose adjustments necessary in special populations, development strategies, whether to accelerate a development program or to potentially take more time and study earlier clinical objectives rather than moving forward to later stage. From there, understanding that question both qualitatively and quantitatively, we can think about the relevant knowledge sources and knowledge gaps that we're faced with in making a decision. Those knowledge sources can be data that are derived from internal proprietary sources, maybe public registry data for a particular disease area, competitor metadata from the literature. They could be pre-existing models for pharmacokinetics, pharmacodynamics, patient population distributions, competitor response, and other sources of information. Any gaps in the necessary knowledge might be filled by actually building models or bridging across data in a quantitative way. Or it might be the case that a new experiment is necessary and additional data should be generated. All of that then is incorporated into a model that is fit for purpose to simulate the scenarios in question. Those simulations are then evaluated in the context of decision criteria. They could be criteria around cost-benefit trade-offs around the therapeutic. And they could also be considered based on different criteria depending upon the stakeholder position. Patients in an unmet need for a new therapeutic may have a different point of view than regulators who might be risk averse and cautious about getting a new therapeutic approved too rapidly, for example. Once those simulations are conducted and evaluated relative to the decision criteria, then it's necessary to check assumptions. Assumptions are made when we fill knowledge gaps. They're inherent in the simulation problem sometimes because we're typically applying knowledge to a new condition, something where we don't have all the answers already, and we're looking at the potential opportunity to inform that new situation. There are assumptions that are made both explicitly and sometimes implicitly. And also there are uncertainties about some of the model components, the model parameters, and the ability of that model to be useful in guiding the decision for the particular question in mind. Given that, assumption checking, a decision is driven by selecting one of the scenarios or options with the highest probability of meeting the stated goals. Those conclusions can be evaluated versus sensitivity to assumptions and uncertainties, but typically that allows the decision makers sufficient guidance to move ahead with some sort of a decision. This entire process is repeated then as new data evolve, as the development scenario evolves over time, or as we approach new questions. So clearly there's a lot to this concept of model-informed drug development, and to implement that sort of an approach is not a trivial task. There are some benefits and challenges for organizations who aim to make decisions based on model-informed approaches. On the benefit side, these approaches allow us to integrate data, integrate knowledge, and also transparently define assumptions. Knowledge gaps can be bridged as well here too, whereas individual data sets might not tell the complete story. Our theoretical understanding in a model-based approach can bridge those and benefit from the variety of information sources available. MIDD approaches encourage multidisciplinary team input. In fact, it's really a requirement to make sure that the questions are well understood, the quantitative criteria, the key assumptions, and the outcomes that are desired. This is a multidisciplinary team effort. As I mentioned, these more objective approaches mitigate some of the biases and challenges in the psychology of group-driven decision-making, and in the end, allow us to efficiently make use of data to inform decisions in an accurate and sometimes an expedited way. This is balanced with a group of challenges as well. As I said, model-based approaches frequently rely on strong assumptions, and that is potentially a challenge if those assumptions are not validated or the sensitivity of our conclusions to those assumptions are not considered. The analyses are more complex. I showed you a couple of examples where we had simple model-based point and interval estimates that were used to drive decisions. The full MIDD strategy that I just outlined is surely more complex than that, often includes multiple models, different levels of statistical variability and hierarchy, and potentially hundreds of different simulation scenarios. The other challenge is that MID approaches clearly suffer from a lack of proactive planning. This is an effort that requires a lot of forethought and ensuring that the correct data are collected and the appropriate modeling and simulation strategies are developed. For an organization, this also requires an investment in infrastructure and skills to do so. And probably the most difficult challenge is that legacy decision-making habits and processes in many organizations are not typically comfortable or familiar with MIDD approaches. And sometimes there's an effort necessary to overcome or to coordinate those legacy processes with a more quantitative and more complex MIDD approach. We'll review all of these benefits and challenges as we move forward in illustrating the MIDD strategy in the next training module. So I've shared with you some references on MIDD and the psychology of decision making. I'll end with offering that you may contact me if you have questions on this material and reach out to me directly. Thank you for your attention and look forward to our next module.